Hey, dorks! Welcome to another dorks event. This week's theme is microbes, and we are super excited about talking about microbes. We have two awesome microbe enthusiasts here to chat with us. If this is your first dorks, then let me give you a quick rundown of how this event is going to work, and then I'll introduce my co-host and our speakers. So the way this works is we have one person give a 10 to 15 minute presentation. Then we open up the floor for questions, and you can submit whatever question you would like to ask our speakers through the Q&A feature down at the bottom of the screen. So you can submit your questions during the presentation or while we're speaking with the guests. And then after we've done about 15 minutes worth of Q&A, we'll move on to the next speaker. And we'll do the same thing. And then we'll open up the floor at the end for Q&A for both of the speakers if you have any sort of microbe or science related questions that apply to both speakers. So please do feel free to submit your questions. We're excited to have them. And so I'm Kelly Weinersmith, one of your co-hosts. This is my daughter, Ada, who's joined me for some of the events. And my other co-host is Scott Solomon. So welcome, Scott. How are you doing?
Hey, Kelly, I'm doing great. Yeah.
Awesome. So before I introduce our awesome speakers, you and I always share a drink themed for the event. So today it was microbes, and you pick our drinks each week. And so how about you start by telling us what you're drinking?
You know, I have to say that, so when I'm choosing drinks, I end up Googling things like, whatever the topic is, plus the words cocktail or mocktail. And if you start Googling words like, microbe and cocktail, you don't get what you're looking for. If you've worked in a microbiology lab, you probably know that that means something else. Or, if you start looking up like bacteria cocktail, you get all different sorts of recommendations for like treatments for infections that you can use different combinations of antibiotics and drugs. So it was a little bit of a challenge to find what I was looking for. And then I remembered, oh, wait, kombucha, that's like a drink that's supposed to have some beneficial microbes in it. So, what better way than just like drinking a bunch of bacteria, right?
Totally.
And I have to admit, so I've never had kombucha before. So this is my very first kombucha experience. So anyway, yeah, we always do a cocktail and a mocktail. The thing is that this time it was so tempting that I couldn't decide. So I went with both.
Excellent. Well, let's start with the one that I can't weigh in on. So I didn't do the cocktail this week. And I don't know if your first interaction with kombucha should include vodka. That maybe sort of is going to color it. So I'm glad you have the mocktail too. So what do you think?
Yeah. All right, let's go for it. So I'm gonna start with the cocktail, since that's the one I had promised to have.
Okay.
You know what? That's actually pretty good.
Oh yeah.
Yeah, it is. It's the kombucha mule. So it's like a Moscow mule, but with kombucha instead of ginger beer. That's really tasty. I don't know if putting vodka in like just kills all the bacteria and completely eliminates like any potential probiotic benefit, but it tastes good.
But Mark is shaking his head no, like those bacteria are still going. Is that right, Mark?
I think you need to figure out how vinegar is made.
Oh, because
in fact, lots of bacteria use ethanol.
Good point.
Yeah.
I figured maybe we're selecting for vodka resistant bacteria.
I am all the time in my gut.
I'm surprised you like kombucha the first time around. So I've had to sort of become a kombucha lover. The first couple of times I tried it, I was like, I'm not sure why humans do this to themselves.
Yeah.
But over time, it's really grown on me. And now I totally love it. And it's my like go-to drink of choice in the summers after I've done like a long day bush hogging the fields and stuff. I love a good kombucha.
Kelly, I can change your life right now.
Okay.
You're familiar with kefir, right?
Yes. Or except I call it kefir. Am I thinking of something different, or is that a different thing?
No matter what I say, it's wrong. Let's just deal with things that way. And what I discovered a long time ago is that you can passage it through grape juice. And after about 5 or 6 cycles, you have effervescent grape juice that is made with kefir grains, which is absolutely fabulous. Just saying.
Wow.
That sounds awesome. Sounds like a life changer. I'm going to have to try it.
I told you.
Thank you. I appreciate it.
Annelise, you too.
Are either of you drinking a kombucha-based drink today? Or I know Ada's drinking water. Nothing wrong with drinking water.
I'm calling this water.
All right. Whatever you're drinking is appropriate.
Yeah, I've got my milky tea. So
milky tea is good too.
Excellent. Yeah. Excellent.
And so I forget what the name of the mocktail was. I'm looking it up. Ginger lemon kombucha. So it's 4 mint leaves, a squirt of lemon, and some ginger lemon kombucha, and it's great. I don't actually know that adding the lemon added too much to it. I like the mint leaves. That was good. But yeah, I'm digging it.
Yeah, I put more mint in than the recipe called for. I really like mint, and I'm digging it. Yeah, it's tasty.
All right, and our guts will maybe thank us. Although one of the questions that I'm looking forward to asking the speakers maybe at the end is how overblown are some of these claims about how things like kombucha or the the pills you can get with probiotics or whatever, how much those help. But anyway, we can get to that. So let's start with our awesome talks. And so today we've got Mark and Annelise. Mark is going to go first. So I'll give Mark's full introduction first. So we've got Mark's going to be giving a talk. And if you want to start pulling up your slides, Mark, it's a Germ Eat Germ World. Bacterial predators are everywhere. Mark Martin often goes by Doc. That's what his students call him because he has 7 pairs of Doc Martin boots. I went through a Doc Martin phase where I had not quite 7 pairs, but a lot of pairs. And to be honest, I'm not sure why I got out of that phase because they're great shoes. He's an associate professor in the biology department at the University of Puget Sound in Tacoma, Washington. And his fun fact is that he has written and published science fiction in my odd past. So is this like space science fiction or what? Yeah. Can you tell us a little bit about the science fiction you've written?
Well, I've written a few things a long time ago. I used to work in biotech in San Diego, and it was not a fulfilling career for me. And I have many friends in the writing business, and I had the opportunity to write a couple of novels in Larry Niven's universe. And so I was able to put my spin on to some of the Man-Zin Wars novels. So I did 2 of those. And I've also written some ghost stories because I really enjoy supernatural fiction. But I've been a little busy in academia since.
Yeah, that happens. That happens. But oh man, that's awesome. I'll have to read one of your books. We had a section on ecologically oriented fiction where we were talking a bit about the process of writing fiction. We should have had you on that episode also. But all right, I will stop talking and we will watch your presentation. And I'm excited. Although it looks like right now I'm seeing everything that's on your screen. So I'm also seeing your notes and the next slide.
Yeah, it's in presenter view currently. So you might want to see if you can do swap displays.
Uh-oh. So let me see. There were 2 things up. Thank you for letting me know that. Mm-hmm. Everything is kind of an interesting journey, isn't it?
That's life. Yeah. I feel it doesn't matter how many times I've done stuff. It is always a surprise when I try to do it again.
Is that any better?
That's the same. I think if you look at the top left where it says swap displays, I think that might do it.
I'm looking for that now.
Just above, or it may,
I mean, I have that.
Yeah, see, I think you're seeing the full screen and we're seeing the presenter view.
All right, let me try it a different way. And I'm sorry that you all have to deal with this.
Yeah, it's all good. This is just the nature of Zoom, sharing lives over Zoom.
You would expect that I would be okay with that at some point. And now it's not giving me you can't see this now, of course. Now, why is it? Oh, you know what this is? Because I didn't originate this. That's the issue that I am dealing with.
Oh, I'm
okay, I'm starting to understand. So please be patient with me, friends.
No worries.
No problem at all.
Okay, let me see about this now. And
I think that's going to be better. Yep.
How about that?
Yep, we got it.
Yay! All right.
How many PhDs does it take? That's the real question, right?
As many as you have.
I have to
every time.
I have to tell you, we did this once with 4 of us trying to figure out how to get a microscope to work in a teaching lab, and it was not plugged in. So that's important to keep in mind. That is a true story. Hi, folks. I am here to talk to you a little bit about predatory bacteria. Thanks for your patience as I go through this. I also spend some time doing a certain degree of art with luminous bacteria, and you can see some of it over here on the one side. I also have some interesting stuff about my department. I have kind of the logo for my laboratory group and something that Johnny Chang made for me at ASM a few years ago. Let's go ahead and get started. I have a microbial perspective on most things. That's just who I am, friends. And my wife, Jennifer Quinn, who's a mathematician and artist, made me this from Dr. Seuss because I loved Dr. Seuss growing up. I had Maggie Middleton make me this piece of art for when I was given a really wonderful award from the ASM, and I was giving a talk about how microbes are important to my life. But it all comes down to this. If microbial centricity is wrong, I don't want to be right. And it's really true. First evolved, last extinct. And our major problem is eukaryocentrism, and I'm here to change that for you. I have a microbial bias. I'm upfront about my biases. And this is from Pliny. And Pliny wrote here in Latin, I'm not going to say it for you, that nature is to be found nowhere more than in her smallest creatures. And if you see that, then you're not surprised to see this. And that's just how I live my life. Now, what I'm going to do today with all of you is I'm going to give you a little bit of a roadmap of what we're talking about. I'm going to talk to you a little bit about some original general concepts about bacterial predation. I'm not going to be terribly detailed. I'm happy to answer questions about that, but I want to give you the view from 35,000 feet. I'm going to give you several specific examples. I'm going to give you applications to the everyday world. And then finally, I'm going to brag on my undergraduate students. I'm at the University of Puget Sound that does not have master's or PhD students, so all my work is done with undergrads. And with that, friends, and by the way, if anyone needs to break in, I don't have chat up, but I believe a little red dot will come up if anyone does. I do want to warn everybody, the 3 most important words in science are I don't know. And I cannot tell you the amount of trouble I have seen myself and other people get in being unwilling to say it. I travel light. It's easy to say I don't know. And actually, science progresses by these rules. Stage 1, that's a silly idea, and you're silly for proposing it. Look around you. You've seen this over and over again in science. Stage 2 is it might be true in the laboratory, but it's not relevant in nature. You've seen that as well. And you've seen stage 3. I said it was a good idea all along. And one of my research students, who's a wonderful surgeon now in Los Angeles, came up with a 4th stage, and I came up with the idea first. And I have to tell you, this happens over and over in science, and it's going to happen with predatory bacteria, following the examples that we've seen with bacterial viruses or bacteriophage. I want to give you a thought today to start with from Jonathan Swift, because I'm at a liberal arts institution and I love literature. And Jonathan Swift wrote this, which is just well worth saying out loud. So naturalists observe a flea has smaller fleas that on him prey, and these have smaller still to bite him, and so proceed ad infinitum. Now, Swift was talking about literary editors, but it's also true for the microbial world. And it's so funny to think about the fact that there are bacteria out pursuing and consuming other bacteria while we speak. With that in mind, what is the goal of a microbe? It's to make more microbes. And I want to bring up 2 concepts, the one of autotrophy and heterotrophy. An autotroph gets all of its carbon to build everything about a living organism, ranging from proteins to nucleic acids or even lipids on carbon skeletons. An autotroph gets those from carbon dioxide and generates its energy without the use of breaking down organics. So think of the way that plants can use sunlight, although what the plants are using are bacteria that live inside them that we call chloroplasts. On the other hand, how we get our carbon and how we make our energy is to break down more complicated compounds. That's called heterotrophy. And so if you think about it, it's kind of hard to be an autotroph. But if you're eating other things, that's a little bit easier. So what is one excellent strategy for obtaining energy and raw materials under conditions of low nutrient availability? What can you do? Why? You steal it. You steal it from other organisms that have already created biomass. They're a source of energy and they're a source of carbon. And we can call that predation. Now, I find that people have trouble with that on a microbial level where they have no trouble with microbes. So that's, once again, it is like a cruel form of bias against the microbial world. With that in mind, I want to talk about some basic predatory strategies that you see among microbes. And specifically, I'm talking about bacteria with you today. And one interesting approach is called, and this is in great deference to the people at the University of Michigan who came up with this idea, and it's called a wolf pack predatory approach. And the myxobacteria do a lot of this. And what you're able to see here is you have these individual tiny spores germinate, the bacteria emerge from their spores, and they work together, preying upon other bacteria, not one another. And then when they run out of food, they create a structure in which they make more spores for dispersal. So again, these are social bacteria that work in groups, and they generate enzymes in front of them that break down any microbe in their path. Hence the name wolf pack. And it's not terribly specific. So it goes after pretty much anyone in its pack. And you can find anyone in its path, I should say, apologize. I'm used to a mask these days. In any event, this is kind of how it works. But I wanted to show you what you're looking at here, friends, is a colony of E. coli here, typical lab rat bacterium, and a colony of Myxococcus here. And watch what happens. Come on. There we go. You can see the Myxococcus starting to invade the colony of E. coli. Now, these bacteria are working together. You'll start to see ripples as they do. And they're eating the E. coli. And they're proceeding across that rich group of prey organisms, the E. coli, with these ripples. And then pretty soon they've taken over the entire colony. And I want you to note what happens at the end of that period of time. They're still looking outside, but there's nothing to eat. And you'll see these strange structures forming in the middle of the now consumed colony. Very similar to how things started. And what you're looking at are making more fruiting bodies. Those are the tall structures on which new spores are for proper dispersal. So these are extremely common in nature. You see them everywhere in soil. And that's how the wolf pack works. Let me see if I can get past that. Now, the late, great Lynn Margulis wrote a wonderful paper in the Proceedings of the National Academy of Sciences in the 1980s that basically gave 3 other mechanisms by which one bacterium can consume another. Now, many types of bacteria are called gram-negative. They have 2 basic membranes. And in between that membrane is a space that we call the periplasm. And that's where some organisms live that prey upon others. The specific one is one I spend some time in my lab working on, called Delovibrio, which means curved leech. And what it does is invades that space between the inner and the outer membrane. For Halloween, we have this excellent example called Vampirococcus. They're epibiots in that they cling to the outside of the organism and elaborate structures that are able to use the biological materials within. Now, I should tell you that nobody has really grown Vampirococcus because I've been interested for decades in this. But even more so, this organism that's only been described microscopically, it's never been cultivated, Daptobacter, where the organism directly invades. So we have periplasmic, epibiotic, and direct invasion. Of all three of these, the only one that gets studied closely are periplasmic and perhaps some work with a related organism I'll bring up in a minute. But they're everywhere in nature and vastly understudied, just like the majority of living things. So if you look at the one that I've studied, Vibrio, which we're looking at here, you have individual highly motile cells. They impact a prey organism. They invade the region between the inner and the outer membrane. They begin to grow into a filament, septate, and release. I draw your attention to there's a life outside the cell and the life inside the cell. And how they know they're on the inside is a fascinating topic for people like me. In addition, you will be alarmed to learn that once one Vibrio enters a prey cell, it undergoes changes. It rounds up, in fact, and no other Vibrio can invade, which is fascinating. How it really looks under a microscope from work I did many years ago with Tom Pitta is like this. You can see the Vibrios trying to invade, and you see an invaded cell here. If you'd like to cue the theme to Jaws, here's a mass culture, and you can see this poor organism that has a whole bunch of Vibrios trying to get in there. But I think Liz Sockett's lab does a good job of showing you a little bit of what's happening, and Liz is a real expert.
Before we start the movie, we see a tiny black Vibrio predatory bacterial cell inside a bigger fluorescently labeled prey E. coli cell. The Vibrio has invaded this cell, and as we start the movie, we see the Vibrio elongate bidirectionally, growing and using the insides of the prey cell. Then it septates unusually to give 5 progeny cells, and the E. coli goes dark as those progeny cells lyse it and escape and go on to attack more prey bacteria.
Isn't that wonderful? And in fact, my wife put something together for me to please me. Here, check this out. Sorry about the music. So what you're seeing there are some curious things. Years ago, I had a student who created this view because when Vibrio consumes prey organisms, some people used to think it incorporated part of the prey organisms into it. And this student created kind of a Borg out of Star Trek for that one. And you will be assimilated. Their job is to take up and use compounds that the prey organism has. Now, currently in my lab, what we're interested in are global regulators. And this basically means how Vibrio knows that it's inside of the cell as opposed to outside. And I will warn you a little bit about this, that Vibrio is not easy to work with because it will only grow on other bacteria. You can't grow it by itself. I'm also interested in the role of the prey cell cytoskeleton, that is to say the shape of the organism. And I have some evidence that suggests that that's important for prey preference. Finally, well, next to finally, Vibrio has a very unusual set of genes involved with metabolizing amino acids, and we're looking into that. It's very interesting to me. And we've gotten quite interested in things involving sugar metabolism in Vibrio, even though Vibrio should not be using sugars. It still makes the enzymes for that. So I just wanted to give you a taste of the kinds of things that we do in my lab currently. Many other people, at least 4 other groups, look at more of the actual mechanisms of invading cells, which is really quite interesting. The other organism that we study in our lab, and we're not exactly sure whether it's an epibiont, though I suspect it is called Ensifer, which means sword bearer. And this was discovered in the 1980s by Lester Casita. He used a common soil bacterium, which is yellow, called Micrococcus luteus. And he put it into soil samples, and he found in some soil samples that Micrococcus quickly vanished. It was being consumed. And he found that one of the bacteria responsible was what he named Ensifer. And I would say to you for your take-home lesson there that predatory bacteria are literally everywhere if you go to the trouble to look, just like that slide with the stages of scientific research. And here's a video I made in my lab. What you're looking at is Micrococcus, and I put a drop of Ensifer right here. Now, this takes 3 or 4 days, and I used a GoPro for this. And you can see that the Ensifer, which makes this goo called exopolysaccharide, spreads along where the Micrococcus is. It does not do this if the Micrococcus is dead. It does not do this on E. coli. We call this process tracking, and we think it's important for how it identifies prey. Should also point out to you that Ensifer is very specific in what bacteria it eats. I've only been able to get it to eat Micrococcus. We also have had some fun in the lab generating variants of Ensifer that swim faster or slower, and you can see that. The normal one, the ones that swim faster, the ones that swim slower. And we have some evidence that there are differences in tracking activity. I don't want to take up too much of your time. I just want to point out this was work done by a first-year student in my laboratory. We were actually doing in vitro evolution just by finding things that swam faster or slower. And then later, we did some genome work with Ensifer, and we found out a lot about what's going on with the genetics of this organism. And currently, I'm involved with trying to find out what genes are important for tracking and how they're actually breaking down Ensifer and adhering to it. So it's a really interesting question, and I should say attacking Micrococcus. So what's next? Collating mutations that we make in the lab to the genome. Is that goo, that exopolysaccharide, necessary for tracking behavior? We don't know. Remember, bacteria have other ways of moving than just twirling a flagellum. They have fascinating processes called gliding motility that we think is involved here. So we don't know that swimming motility, which is the whirling of the flagellum, is what's behind this. And I'm very interested in groups of these things together ecologically, and how it changes population structures in the soil. But why should you care? This is great for me in the lab. I enjoy it. But one of the biggest problems that we deal with all over the world is the rise of antibiotic-resistant bacteria. Why is this happening? Because the antibiotics don't change, and the hand of Darwin is on all that lives. And what this means is that it's easy for a bacterium to become resistant to an antibiotic because the antibiotic doesn't change. And that's why a lot of people, and this is an open access article that I recommend, are starting to look at the idea of predatory bacteria as living antibiotics. To be fair, we don't know what they look for in prey organisms. Like Delavibrio is pretty cosmopolitan in its appetites, but Ensifer is quite picky, for lack of a better term. You should also know that this idea about using predatory bacteria is a lot more than health. It involves agriculture. Easily 80% of the antibiotics made are used in agriculture, which surprised me. And Maren McKenna has a wonderful TED Talk on the rise of antibiotic-resistant pathogens that I really recommend. If you want to know more about predatory bacteria, I recommend these 2 open access articles here. One is from 2007 and the other's from 2016. I don't suggest the one that I wrote a million years ago. But what's interesting here is you get some really wonderfully well-written ideas that are well worth your time. So this is like something that no one thought. They thought it was kind of a curiosity, but the more we look, the more we see it. And I'm starting to think it'll be applied to health treatments pretty soon. So summing up this business, predators are very common in nature on a microbial level. And there are many strategies that they use. The wolf pack is not specific. Ensifer is only interested in one thing, which is fascinating to me. As I say, some predators are picky and others have more cosmopolitan tastes. Some have specific techniques to invade, like Delavibrio. It uses fascinating type 4 pili to get into that periplasm. And then we have the hydrolytic enzymes secreted by the myxobacters. And I believe, like bacteriophages, these bacterial viruses that are all the rage now, predatory bacteria will likely have ecological impacts as well as what you see in a medical setting. So they're really excellent candidates for dealing with potentially disease-causing bacteria. And I'm delighted to see that work starting to progress. So just a hat tip to my students. I call my students Doc Martians because we think that's funny. And it's very much like a boot camp for graduate school. I give students their own project in my lab. We work together. We make things happen, both during the academic year and the summer. I will say that publishing is far too slow, but I train scientists. That's what I do. The way in which they approach problems is the most important thing to me. It's about community support and critical thinking. So where I do research here, this is a picture in my cluttered lab. Again, we don't have MS or PhD students. We have standard equipment, but not super fancy stuff because we're a small institution. And I generally have 4 students during each semester, usually 3 students in the summer. And we do a lot of stuff in the lab that we have fun with, as well as the work that's research oriented. So the results speak for themselves. Of the students who spent more than 1 year in my lab, 6 are faculty members at various parts across the country, including one person who just got tenure at Stanford. 25 have or are in PhD programs, 5 in MD programs or have their MDs, 3 in PA programs, 5 are wonderful research techs. And so I preach the one microbial truth, first evolved, last extinct, as much as possible. But what I really am, friends, is a talent scout. That's my job at the University of Puget Sound. And I'm good at it, and you won't hear me brag on myself beyond that. So I know I've gone quickly, but I wanted to give you a taste for what I think about predators and the wonderful students that make it possible. And I want you to remember that we are part of a quality quorum here today. Bioluminescence only takes place in groups, and you're making me feel bright today by being here with me. I want you to always be brave, to never give up, and be true to your passions. And I want to thank you all for this opportunity to chat. Thank you so much, everybody, and I'm happy to take questions whenever.
Yay, that was awesome. I feel inspired, and I have tons of questions now about bacteria. I could have listened to you talk about that for hours. And so let's see. All right, where do I start? So you had mentioned, you sort of started to address this in your closing statements. What does a predation event look like? Are they like battering, ramming their way in? Are they secreting chemicals? Do any of them bite? Is it like phagocytosis? Is it all of those things? Tell me more.
Do you sense the sweet, sweet microbial tears on my face?
I do.
All right. The answer to your question, and I love doing this in class when I'm given a lot of things, is I'll say yes to all of them, because that does take place. Let's talk about Della-Vibrio for a minute. The space between the inner and the outer membrane is literally 40 nanometers wide, okay? Della-Vibrio shoehorns its way in there. It does this, and this is Liz Sockett's work, using what's called a type 4 pili system, which is fab. I want you to imagine a super duper shoehorn. And that's how that works, to slip into that space. Compare that to the way the myxobacters work, where they simply secrete a ton of hydrolytic enzymes in front of them, breaking things down. We think that Ensifer is an epibiont. There aren't good micrographs of it because when you grow it, it has all that goo. But Lester Casita found some wonderful EMs that he took in the 1980s that looks like there's this rod sticking out from it, which is why he named it Ensifer. And what's interesting to me is that Ensifer is closely related to the fast-growing rhizobia that form root nodules on leguminous plants. And we're fascinated by that. There are no genes in Ensifer involved with that symbiosis, though. So it may be this is just a common thing in the soil. You know, I need the equivalent of nature red in tooth and claw when it comes to microbes. Would that be like red in Pilis in Fimbri? I don't know, but I need something like that.
If anyone's going to figure out what that is, it's going to be you. So that it sounds like you're onto something. And I'm sure we'll all be saying it in a couple of years.
You know what I say about myself, and I really believe it, and I can see Annelise already agrees with me. I'm a giant goofball, but I'm microbiology's giant goofball.
That's the way to be. We're all goofballs here. And,
yeah.
and if you don't geek out about your stuff, then it's, hard to go to work every day.
Mm-hmm. And I haven't even talked about the awesome stuff other students have done in my lab. This is just one piece of it.
I love it. I love it. Mark, that was great. And you've helped me to expose my own eukaryotic bias, despite me thinking that I really was, more open to the prokaryotic way of seeing the world. But what I had always thought was that predation is something that first came about like in the Cambrian with the first, multicellular predators. And you're making me realize that's not true at all. Yeah. That predation is something that prokaryotes have been doing for far longer. But then it made me wonder whether we know or have a way of knowing when that first evolved. Like, was predation something that was present at the very beginning of prokaryotic life, or do you think that predation is something that came about later once there was some critical mass of different types of prokaryotes?
I have thoughts on this, but obviously I don't have a time machine to go back and look. But prior to the great oxygenation event, when the ancestors of cyanobacteria could fuse photosystem 1 and photosystem 2, and this changed the face of our planet. It's the green marauder, the green plague, generating all this oxygen. I think prior to that, biomass was really quite restricted. And there's evidence for that in the geological record. And I think under those conditions of what I believe I can reasonably call oligotrophy, I think that there would be a strong evolutionary pressure toward being able to prey upon other microbes to gain your what you need to live. I want to say parenthetically, if you've done any reading about the Asgard microbes, I don't know if you have, there's a lot of evidence that those early archaea actually had the equivalent of cytoskeletal altering proteins. So they may well, taking you back to your question, Kelly, have done the equivalent of phagocytosis. We think that's how mitochondria were first incorporated into eukaryotic cells after all. But to me, I would say that predation, because it's all about making a living, and those that are better at it make more of themselves. So as far as you being worried about having a prejudice or a bias, I call these the 3 deadly centrisms. And the first is eukaryocentrism, which is in my first year class, they're so tired of me talking about. The second is oxycentrism, because oxygen is genuinely a poison. We've just learned to deal with it. And the number 3 is colicentrism, where we think all bacteria are E. coli, and none of these things are true. We got to look.
I love that. Yeah. No, that's so true. Wow. So Annelise, if you have any questions, feel free to jump in. I also want to encourage the folks that are watching live, if you have questions, to go ahead and write those in the Q&A, and we'll get to as many as we can. But Annelise, did you have anything you wanted to ask?
Yeah, for sure. Well, first I wanted to say that I appreciate your anti-eukaryotic centrism, because I have been involved in a years-long debate with my best friend who does research in mice about whether mice or bacteria are a better study system. And I don't think we're ever going to agree with one another, but...
You need to get a button, Annelise, that says no use. and you need to wear it all the time now.
Yeah. Yeah, so I had 2 questions. Maybe sort of going a little bit further into like biotechnological applications. Because my lab that I work in does a lot of synthetic biology applied to soil microbes. So the first question I had was like, I'm fascinated by this concept of using a mix or like a microbial community as a soil amendment as fertilizer, instead of like a nitrogen-based fertilizer that could cause some sort of pollution. And so I was wondering, like, do you envision any kind of role for predatory bacteria in those kinds of soil amendments for, I guess I was thinking specifically agricultural purposes, but maybe more broadly?
I think that the idea of what takes place in the rhizosphere is an interesting one, or even on the surface of leaves. And there are people working on that. And I don't know the role of predation there. I'm reminded a little bit of what the role that bacteriophages have on populations. They tend to promote diversity, and they tend to keep one particular organism from predominating because they're constantly changing their tropism toward prey. And so I would argue that the role of a predator, when I say argue, that sounds so militant. I would advance the idea that predators actually work toward diversification. And just as we worry about monoculture in agriculture with regard to the plants and crops, I would argue we also have to worry about monoculture in the rhizosphere. I know from because I used to work in Cynorhizobium mellilodi a long time ago. And I have to tell you, I would see people that would find variants that were better at fixing nitrogen in association with leguminous plants. And then when you would drop those into a soil, they would vanish because they couldn't compete. And then people got scared. What if we find competitivist genes? And like I read a lot of science fiction, so that would like occur to me. But I think it's more complicated than that. I think, again, most things in life are Darwinoed by evolutionary processes. And so that would be very interesting to see. But I do think, just as when we're dealing with issues involving in the human gut, it looks more and more like it's community that needs to be introduced rather than one keystone organism that makes changes. Mm-hmm. So in some ways, you might worry if you introduce too many predators, would that have a negative effect? And it might. That's why I would love to see how populations change over time. Do they reach a snowshoe hare and lynx kind of association? And they probably do. But you're never going to see that in a shake flask, right?
Yeah. Yeah. Okay. Yeah, that's really interesting, just because my brain immediately went to using predatory bacteria as like quote unquote kill switches is what we call them in synthetic biology, but like the idea of controlling something that you introduce into the environment with predators. But I did not think about the diversification aspect. So that's really interesting.
I will say that, are you familiar with gasp phenomena?
It sounds familiar, but I don't remember exactly off the top of my head.
So when you starve E. coli and it goes through the typical lag phase, exponential phase, stationary phase, and the slow death phase. Steve Finkel and Roberto Coulter showed many years ago that you have this extended death phase that goes on for years. And within that, you see lots of ups and downs as you get evolution taking place among the survivors. It's like Thunderdome for microbes, if you know that reference. You probably don't, Annelise, but the rest of us do, right? 2 microbes enter, 1 microbe leaves, Thunderdome. And so in a similar way, what I worry about with your kill switch idea is that you're going to get that constant push and pull of Darwinian change. But it is something that could be studied very easily. For example, if you take something that's been in extended stationary culture for a long time and you compete it with, quote, naive bacteria, the stationary culture is always better because it's accumulated mutations that make it better. And it's a really great exercise I have my students think about. So what are they perceiving and what kinds of mutations would give them an advantage? I have to warn you, Kelly, I'm the kind of person, if you ask me what time it is, I tell you how to build a watch. So I'm sorry about that.
No, that's perfect. I personally think it's way more interesting to have like the long detailed answer than, yes, and then no details. And so no, this has been perfect. I do think that probably we're gonna go to Annelise, and then when we open it back up again to everyone, I'm sure there'll be more questions. But that was awesome.
Thank you.
It was, absolutely. Thank you, Mart.
Much appreciated.
Okay, so our next speaker is Annelise Goldman. Annelise is an undergraduate student at Rice University, where Kelly and I both teach. And she is doing research in the Silberg and Masiello labs. And her fun fact is that her parents had to specifically create a rule banning intentionally culturing bacteria in the house because she did it too often, which, because you said intentionally culturing, I'm guessing you're not talking about like, you needed to clean your room more often, and that's what they called it, right? So how are you culturing bacteria at home?
Well, I would like to take the blame off of myself and say that they allowed me to purchase pre-poured agar plates for a science fair experiment, and then it went a little bit wild when we all had to leave campus at the beginning of the pandemic. I went home and wanted to make these really cool way of like exploring soil bacteria and different metabolic processes called Winogradsky columns, where basically like you you have a jar, like you can take a pasta sauce jar and fill it with mud or dirt, and basically over time, because there's an oxygen gradient that you introduce into these things, you can see different kinds of bacteria growing, and so this was after the ban was in place, so I had to do it outside. Wasn't allowed to bring them inside.
After the ban was in place, I love that.
So
you had already done enough experiments. They were like, "That's it, enough."
Yeah,
Your brilliance was not recognized.
Thank you. That's how I would like to perceive it.
It's sort of like signs you might be a biologist. If like, you open the fridge or the, pantry or something, and you find experiments or cultures or specimens where other people might find like normal household items or food, you might be a biologist. Yeah.
Been bad for Zach a couple of times in that regard.
Oh yeah. Same. Yep. I currently have a bird tapeworm in the freezer that I'm probably not going to be able to keep there much longer. It's been questioned multiple times by the family, but
I can't think of a much better use for a freezer personally. I'm wearing my I Heart Parasite shirt though today, but anyway, I hope they let you leave it in there till you find a good use for it.
Exactly. And if anybody out there like wants a bird tapeworm, that's why I'm hanging on to it. So anyway, I digress. Annelise, we're excited for your talk. The title for her talk is Mining for Genes in a Gold Mine, Exploring Microbial Sensing in the Deep Subsurface. All right, take it away, Annelise.
Cool, thank you. So can you see everything well regarding the presentation?
Looks perfect.
Okay, awesome. Yeah, so thank you, first of all, for inviting me to talk today. I'm really excited to be here and to share my love of microbes with you all. So I realized now that the first part of my talk is very similar to Mark's, so I will try to keep my this is why microbes are cool bit a little bit shorter. But I sort of do research at like the intersection of geology and microbiology. So I think this will be a slightly different perspective. So first of all, I just want to do my quick spiel for why microbes are important. Because even though, especially with the pandemic, microbes have kind of gotten a bad rap, they really shape our daily lives. And here are a few reasons why. So first of all, we owe a lot of our atmosphere's oxygen to marine cyanobacteria. Without them, we would have at least 20% less oxygen in our atmosphere. And also they were really responsible for creating an oxygen atmosphere on Earth in the first place billions of years ago. We also know that gut bacteria are essential to our health, and there's a lot of research right now sort of exploring the relationship between the gut microbiome and other aspects of human health, the gut-brain connection, things like that. Also, we know that these nitrogen-fixing bacteria that live in these little root nodules on legumes, for example, provide nitrogen to our crops, and so they're really essential for agriculture. Also, as we were talking about kombucha, there are so many different kinds of fermented foods that we all love that we couldn't have without bacteria. And finally, we really owe our clean water to bacteria because they're used to help remove organic pollutants and other things that make our water dirty in water treatment plants so that we can have clean water to drink. And in order to do this, microbes use a really diverse set of metabolic processes. And so first I'm going to compare this to what humans do, and then we can see what microbes do and see how it's different. So for humans, for both our energy source and our carbon source, we use food, obviously, and that it basically comes in the form of glucose and other kinds of preformed molecules like amino acids, etc. And we also need an electron acceptor, and this is actually basically what we breathe. So without going too much into how like metabolism works generally. Basically, at the end of the metabolic process of passing electrons from the energy source, we end with an electron acceptor. Ours is oxygen, so that's what we breathe. And bacteria need all of these same things in order to survive, but they have a lot more at their on their plate in terms of what they can do. So bacteria can be photosynthetic, or they can use preformed molecules. So they can use sunlight as their energy source, or organic molecules like we do, or inorganic molecules. So this to me is where it gets really cool, because microbes can use forms of iron or sulfur or hydrogen gas or carbon monoxide or different forms of nitrogen as their energy sources. For their carbon sources, they can use organic molecules like we do, or carbon dioxide like plants do to fix with photosynthesis. And this to me is the coolest part, is that microbes can breathe metal, which is so cool to me. So for their electron acceptors, there's a really wide variety of molecules that can be used, including oxygen like we do, but also forms of nitrogen, forms of manganese, which is a metal, forms of sulfur, forms of iron. So yeah, it's just really cool to see what bacteria can do, and it really allows them to survive in a really wide range of environments, which I'll get to in a few slides. And so really, at least in my lab, we think a lot about electron transfer. And so when we're thinking about metabolisms, especially in relation to geology, it's really all about the electron transfer in terms of taking an electron from an energy source and eventually passing it to an electron acceptor, what the microbe quote unquote breathes. So I think Mark touched a little bit on this also, but bacteria can live in communities. In fact, they basically always live in communities as far as I know. And these are called microbiomes. And so communities can be either one species or multiple species, but I think the multiple species ones are really cool. And they can either be fixed on a surface or free floating. So often when they're fixed on a surface, they exist in what's called a biofilm. And it's basically like living in a kind of jelly that the microbe produces. And this can involve different species collaborating with each other. Maybe the waste product from the metabolism of one bacteria can be the energy source for another bacteria, and so they can share molecules that way. But they can also compete against each other and produce antibiotics, toxins, etc. In these biofilms. And here you can just see a microscope image that's false colored with the different shapes of the bacteria and different kinds that can exist in this biofilm that's fixed on metal, or even things like your teeth, like dental plaque is a biofilm, and so on. You can also have this planktonic or free floating form. Here you can see some nutrient broth that has no bacteria growing in it. And here you can see nutrient broth that has bacteria growing and it turns cloudy. And those are all free floating in the broth. And the way that we often study these communities is with a suite of techniques that we generally call omics. But the technique that I use that I'm very interested in is called metagenomics, where basically you can collect genetic information from the entire microbial community. So essentially you take a bit of biofilm or a liquid sample, for example, and lyse all those bacteria, make them break apart, and then take the genetic information, the DNA from that entire community, and sequence it to just understand in general what kind of metabolic processes are happening, what bacteria are actually present, and then you can assemble those and really understand for individual microbes in that community what's going on. And finally, getting into more of what I do research on, microbes can live in extreme environments. They can live basically anywhere. So some examples include hot springs. These go up to like 100 degrees Celsius, this one, which is boiling, at Yellowstone. So microbes can live there. They have also been found in the sea ice in Antarctica. They can be found in environments with very low pH, so very acidic environments like this acid mine drainage stream. And then they can also be found in ocean hydrothermal vents, which is basically this structure on the seafloor where there's some magma kind of starting to bubble up from the inner part of the earth, and that releases all of this sort of mineral particulate, very sulfur rich, just you wouldn't really think of this as being a habitable environment, basically, but it's super hot, really weird compounds being emitted, but there are still microbes that survive here and make a living in this extreme environment. And finally, what I study is the terrestrial deep subsurface. You can also consider the deep subsurface underneath the ocean. But basically what this means is that if we think about the different layers of the earth, we sort of start with soil at the very top, and we know microbes are there. And then we go into some sediment, then some sedimentary rock, and finally we get to igneous rock. And igneous rock is not entirely solid. It contains some fractures. And those fractures contain fluids that have basically drained over a really long time from the surface into the fractures. And bacteria can live in those fractures and also on the surface of the exposed rock, so that as biofilms on the rock as well. And this can, life has been found down to at least 2.5 kilometers below the Earth's surface. I'm sure more than that, but the most recent paper I read happened to be 2.5 kilometers below the Earth's surface. And that's just kind of nuts to me that life can survive in all of these strange places. And as a side note, like, it's interesting to study this, not only to understand just how life works, but also to understand how life might have formed in the first place. A lot of these are extreme environments that we think the early Earth could have been like, or that a place like Mars could be like, where we sort of want to explore if life could form on other planets. And also it's interesting from a biotechnological perspective. I think the most prevalent or applicable example is that so we're all familiar with PCR now that the pandemic is a thing, and we have those PCR tests, but the compounds, the molecules that are used to make PCR happen were actually discovered in microbes from a hot spring. And so that particular enzyme comes from here, and it really wouldn't have been possible to develop a process like PCR without studying these microbes. So it is really important that we understand how life is surviving in these extreme environments. And one of the things that we need to understand to do that is how microbes sense their environments. So this is sort of in general what my lab is interested in. And we know that microbes need to sense their surroundings in order to survive. But something that we don't have a great understanding of is how local environment might influence what a microbe senses. So here you can see some examples of what we imagine microbes need to know about their environments in order to survive. And we can break it down into abiotic, so non-biological factors, and then biological factors. So talking about the non-biological factors first, we can think about molecules that are relevant to redox processes. So basically molecules that are relevant to the transfer of electrons. And as I pointed out earlier, the metabolic processes that we think about for microbes that are associated with rocks really depend on these electron transfers. So when we're thinking about geomicrobiology, this is really what's relevant, or one of the most important things. We can also think about other kinds of nutrients that are available in an environment, and also the chemical gradients that all of these compounds exist in. They might be really concentrated in one place, not so concentrated in another, and a microbe needs to sense where that concentration is in order to move towards those, to get those relevant molecules. We can also think about biotic factors like cell-cell communication between microbes, then plant-microbe communication, toxins and antibiotics, and also viral infections from phages. And in order to do this sensing, microbes use a protein called a histidine kinase. And so this is the protein that I study, and it consists of 3 relevant parts. The first, that's kind of the most important in terms of being able to do the actual sensing is, of course, the sensor domain. And this is what is tailored to look into the environment and find one specific stimulus. Then there's another domain that's on the inside, and this is highly conserved, so very often found between all different kinds of histidine kinases. So the sensor domain will vary between all of them based on what it's sensing, but this HATPase domain, which interacts with another protein, will be very highly conserved, so found in all of these proteins. And so the HK interacts with another protein to then cause some sort of change in gene expression upon sensing the relevant stimulus. And what's also important to note is that up to 2.5% of a bacterial genome has been found to encode these proteins. And so we know that they're extremely important to be found in such a high percentage in microbial survival. So I'm studying sensing in this really cool study site called the DeepMine Microbial Observatory. It's an abandoned gold mine in Lead, South Dakota, that's been converted into a study site for microbes in the terrestrial deep subsurface. There are 6 different boreholes that have been drilled going as deep as 1.5 kilometers below the surface of the Earth. And each of them is drilled into a different kind of rock. So they're what we call geochemically distinct. And basically how this process works is that our collaborators, sadly, I didn't get to do this, I would love to do it, but our collaborators have gone and drilled these boreholes into the walls of the mine. And I like to think of it similar to tapping a tree for sap, is basically you drill into the wall of the mine, or in this scenario, the tree, and what you get out is either syrup or these fracture fluids that contain bacteria and other kinds of microbes that are living in the fracture fluids in the wall of the mine. And so what our collaborators have done is collected the metagenomic, so genetic information of the community living in this fluid, and also geochemical information about the fluids themselves. So in order to really understand how microbes are surviving in this very extreme environment, we also want to understand the geochemistry of the rocks that they're living in. If we can understand more about the rocks, we can understand how the microbes are interacting with the rocks, and so on. And so this is how we're sort of exploring this very low nutrient environment. And we have a few hypotheses about how sensing might change in this very low nutrient environment. So one hypothesis that we have, which has been shown in a marine data set, so microbes from oceans, is that in a very high nutrient environment, which we call copiotrophic, there will be lots of opportunities for sensing, and therefore there will be more sensors in the genome of a microbe. In a low nutrient environment, like the one that we're exploring, we think that maybe the sensors are too energetically expensive to maintain. And so like they require a lot of energy to be kept in the genome, and so they're eliminated from the genome. So that's our first hypothesis. Our second hypothesis is basically the opposite, that in a high nutrient environment where nutrients are plentiful, there will be less pressure to sense their surroundings because there's so much available to them that there's not a high need to be able to detect it. They just need to take it in. And so we think that maybe there would be a decrease in the number of sensor proteins. In a low nutrient environment, we imagine that there might be a need to increase the number of sensor proteins because microbes would need to quickly take advantage of any nutrients that are nearby. So we have these two hypotheses, and we started looking into this data set computationally in order to see which hypothesis might be right. So we constructed what we call a bioinformatics pipeline in order to identify our proteins of interest. So don't worry too much about all the different programs that are shown here, but basically the way that this works is that we get the metagenomic data from our collaborators. And so that's DNA. And then we pass it through a number of different programs that turn that DNA into what the encoded protein would likely be, what that sequence would be. And then we identify those proteins and pull out just the proteins that we care about. So at the end of the day, we are left with a set, basically a list of potential histidine kinases, sensor proteins from the bacteria in this mine. And so what we found is really interesting, really exciting. When we started looking at this data set, we thought, oh, these microbes exist in this really low nutrient environment. They're not going to have any sensors because it's just too energetically expensive. And what we saw was basically the opposite. So more consistent with our second hypothesis. So what we see here is a plot of the number of sensor proteins per 100 genes identified in these metagenomic data. And then on the y-axis, we have the different site that this data was collected from. So we have our two surface control sites in gray, and then these colored violin bars, or violin, yeah. These colored ones represent the different sites, and they're they're increasing in depth. So the deepest one is at the bottom, shown in red. So basically what we can see here is that the median number of sensor proteins actually increases generally with depth, and the range also gets generally deeper with depth. So we see that especially at this deepest site, there are a lot more sensor proteins, and also there can be even up to nearly 4.5 sensor proteins per 100 genes, which is pretty incredible compared to previous numbers that have been established. So we were really excited about this, and we wanted to see how the presence of these sensors, the abundance of these sensors, might be related to the geochemistry of the environment. So we looked, this slide is a little busy, so I'll walk you through it. Is basically we looked at a number of different geochemical parameters or factors that have been sampled at the mine site. So here you can see some of them. Often it's measuring the concentration of different kinds of ions that can serve as electron donors or electron acceptors. So participating in that electron transfer that's really key to microbial metabolism. And so what we found is that there really wasn't a very high correlation between these ions and variability, or sorry, between the variability of these ions over time and the abundance of the sensor protein. So the reason we were looking at the variability of these ions over time is because we thought, it's not going to be the concentration of something at a single time point that a microbe will need to sense, but rather how the concentration of that thing is changing over time. If there's a very high change in the concentration of nitrate, for example, over time, that means that the bacteria will probably need to sense that more than something that's not changing very much, was the hypothesis we went into this with. So we saw there's not a lot of correlation with individual ions, but when we look at this measure called oxidation reduction potential, which we can basically use to represent the ability of the environment to facilitate electron transfer, we see that there's a much higher, not necessarily very high, but greater correlation with the number of sensor proteins per site and the variation, which we're representing with standard deviation. So the variability, or how dynamic this oxidation reduction potential is over time. And you can also see that plot here, how it's changing for each of the sites. And this is over the course of several years. And so basically what we think is happening is that if there's a large change in the ability of the environment to facilitate electron transfer, that means that there's more metabolic processes that are available to the microbe, and therefore there will be more sensor proteins because they need to sense those metabolites, the things that they need to perform their metabolism. So that led us to create an alternative hypothesis to these two that we talked about at the beginning. So basically, we think that it's environmental variability, or how dynamic an environment is, that might drive sensing capability, not simply how abundant a nutrient is. So essentially, we think that when the environment changes often, there's more evolutionary pressure to sense their surroundings. And when the environment is static, there's not much need to sense. And so they'll eliminate those sensor proteins from their genome. So that is all I have for you. I wish I had the time to tell you more about this really cool site. We're also starting to look into the actual sequences of these proteins to really understand them better and really be able to look across scales at how life survives in this extreme environment. But yeah, that's all I've got. So thank you so much for listening. And also thank you to all of the people who've contributed to this project. And that's all I've got. Thank you so much.
Wow, that was fascinating. Thank you, Annelise.
Thank you.
I knew a little bit about some of that work, but that really is amazing.
Thanks.
Wow. So I really like how you highlighted just the fact that microbes live in basically every environment, right? I mean, so it actually got me to thinking, like, do you believe that there are any places on Earth that we can access, I guess, where we won't find microbes? Or is it just a matter of we haven't looked hard enough yet?
I mean, I would be really surprised if there are microbes existing at like the very core of the earth, because
fair
like
point.
How do they get there? And also like it's so high pressure, and very warm, very hot. So but also we can't access the center of the earth. So I'd say, like, in terms of things that we can access, I think that we've found some kind of microbe basically everywhere. I think I kind of focused on bacteria in my talk, but there are archaea that I think can exist in more of those extreme environments than bacteria can. But.
Right. Yeah. Wow. Very cool. Thank you.
I used to say wherever there's liquid water and an energy gradient, that's it.
Yeah.
So I have a friend who disagrees with me and says with deep sea hydrothermal vents, you get water coming out because of the pressure at 300 degrees
Mm-hmm.
C, and maybe that causes everything to fall apart, but I'm not sure about that.
So Mark, does that mean you think, oh, sorry.
No, go ahead.
Does that mean you think there's bacteria on Mars?
I'll bet you money.
Okay.
I think you'll find well, see, first off, I think we're Martians.
I've heard that argument. I'm not sure how convinced I am, but
well, Mars cooled first.
Okay. And so the idea is it got blown off of Mars and came to Earth and seeded us and somehow survived the trip through the vacuum of space, probably for thousands of years. But I guess bacteria are pretty hardcore.
They're metal. And in fact, like, breathe metals, as Annelise told us.
I know. I love that. I actually wrote that quote down. Like, I love that quote, Annelise, that
Thanks.
Microbes can breathe metal. Yeah. They're so hardcore.
I have a comment that I'm going to try to turn into a question in just a second. So my daughter was inspired by your talk. Yay. And she did some artwork. It's microbes can live anywhere. And so
I love it.
In the one with the waterfall, this is like a microbe in like a little water slide, and then this one is a funny one where like there's this really cold mountain and this bacteria is like, ow, hot.
Anyway, so Ada learned something today.
Yay, I'm glad.
So I have this vague memory. So I got a degree in ecology, and there was a grad student at the time who was studying how information impacts the decisions made by prey to like sample and collect data about their environment. And I think the point that they were making was that if there's never any predators around, then you're out all the time. Like, so essentially, like, you don't bother looking for information because they're never there. But or if they're always there, you don't bother looking for information either because you know that they're always there. There's just tons of them. You don't need to sample. But in these intermediate things where there were predators sometimes, but not other times, then the prey species did a lot more sampling of their environment to try to, like, because they didn't have all the information that they needed about the risk. And, Mark has got me thinking that we need to be thinking about microbes as, you know, in a, like the rest of the animals we think about. And so I'm wondering if maybe like information theory that ecologists have done could be applicable to what your bacteria are doing in these. Yeah. And that's maybe more of a comment, but maybe you can pretend I asked a question.
Yeah, well, something that I thought about when Mark was giving his talk was that we're sort of zooming in on some individual microbes in this data set that are outliers, because outliers are always interesting. And one of them is a Vibrio that has a ton of sensors. Like I think maybe around 140, which is a lot. Like E. coli or your standard strain has like 30 to 35. So that I think is really cool and sort of feeds into what you're saying. I don't, like, it makes a lot of sense to me that they would need to sense more, but that is just more of an anecdote rather than a
no, and I think you make an excellent point. Now, for the Vibrio / Vampirococcus, remember, no one's actually grown it. That was fascinating work where they went and fished it out, you know, from a complex population. And it may be that it's not cultivatable under our conditions when all this stuff was originally visualized in the mid 80s from a Meromictic lake in Spain. Oh my gosh, I remember that. These were all EMs. And that's why, you know, I'm not sure. I think there's a selection bias, things that we can study.
Yeah.
And I agree with you 100% that things that are out looking are going to be looking. What I worry about are things where I've been wrong plenty of times. And a good example is the size of Vibrio's genome, which is not small. And you would expect it to be small, but it isn't. And I don't understand that. And, you know, at some point, I just have to throw my hands up and say, I am not wiser than natural selection.
I think I also forgot to say, Annelise, that your presentation was amazing and like way above what I possibly could have handled as an undergrad. Like you blew my mind. That was epic.
Thank you. Yeah. Thank you very much.
Yeah. And then does Mark, do you have a question you wanted to ask?
No, I thought it was fabulous. I put in the chat, you were talking about my favorite thing, Weinergrowski columns, and we did a time lapse I had a student do that you might enjoy. And by the way, Annelise, do you know what mudwats are?
Mudwats? No.
Because Ada needs one. What they are is they're a bacterial battery that you use with microbes. You set it up in mud and it starts to generate electrical current and makes a little light flash. And they're on Amazon.
Mudwats. Yeah, we were doing.
Look at me sadly, Kelly. I'll send you one. But that's good. But anyway, this is what you and I are going to do, Annelise. You don't know it yet, but we're going to set up Weinergrowski columns hacked in from mudwats, so they'll differentiate with the sulfide and the oxygen and also generate current, because I want to see that.
Wait, so do these things, I guess I'm not totally understanding what this mudwat thing is. Is it mud itself?
It's a microbial fuel cell. So you're using microbial respiration to generate current.
Okay, got it. Yeah, for a second I got excited and thought maybe it was cable bacteria.
You might have them in there. I don't know for sure that you don't. You know, because I whenever my students set them up, I call it planet and then the student's name, because that's what it is.
Yeah.
Right?
You guys are now making me think that in addition to sharing cocktail recipes before D.O.R.K.S, we need to like share experimental protocols and have people like do little experiments ahead of time that they can share the results of. That would have been perfect in this case.
You know, Isaac Asimov said a million years ago, the science fiction writer, said that science doesn't progress when, as he put it, a damp Greek emerges from the bathtub shouting Eureka. Okay, making fun of Archimedes, I know. Says science advances when someone says, that's weird. And, and I agree. So having people understand that they can do a lot of science, and they can actually use their own eyes and their own ears and just sense things for themselves. They can set up Weinergrowski columns. It's right out there. And I'll send you guys links that if you want to put up associated with this, because I recommend everyone just start looking around.
Yeah, please do. We'll share that and we'll put it up on our Twitter. Yeah.
This is the second time these columns have come up in D.O.R.K.S. And every time Yeah, the other one was Aaron Reddy, who does planetary protection stuff. He had the astronauts do them.
Right.
And so now I feel like I've heard about it enough times. I need to do it. It's my turn to do it.
The universe is telling us. Yes, exactly.
But Scott Chmielewski, when he was a postdoc for Roberto Coulter at Harvard, he made one for the Harvard Museum that was probably 12 foot tall.
Wow.
And that you don't need to do.
Yeah, I know. I'll start small. Yeah, like an old pesto jar or something like that.
Oh, yeah.
I've got a question for both of you, if that's okay. And so it's sort of a general question. So you both started your talks by saying bacteria either have a bad rap or they're not thought about enough. My sense is that there was a while there actually where they had a really good rap, and I don't know that they totally deserved it. So for a while there, people were like, probiotics and prebiotics, they're going to solve every single problem we've ever had. And they explain everything that we've ever had questions about ever. And so I guess I'd like to hear, do you two feel like there was a period where bacteria were being oversold and it's sort of equilibrated, or are we still maybe riding out that wave? No, Mark definitely has thoughts on this.
Well, since I talk so much, Elise, would you want to go first?
I mean, sure. I don't think I'm as familiar with the entire path of the field. But I don't there is a lot of research right now going on in understanding the human microbiome. And that is sort of related to the probiotic thing, like the idea that if you take more probiotics, you can, I don't know, increase the diversity of your gut microbiome or something. And that would be good somehow. There's talk about like the gut brain connection and this idea that like gut microbes do a lot more in your body and a lot more sort of signaling and controlling your body than you'd like to think. And so I don't know, I think the research is really cool, but also like it sort of needs to be approached with a little bit of caution that maybe not everything is about the microbiome or about probiotics, as you say. Like it's really cool and it might be, but also it's not sort of the end all be all solution to problems. Maybe that's too skeptical. I don't know.
No.
Probiotic panacea. That's like needs to be the name of a like documentary expose or something. Yeah.
Annelise, that's so well stated. I want to openly congratulate you for that. Don't you think that you're too critical on it? Because you need to be. I make jokes with my students that hashtag devil microbes. Yes, that's a tardigrade on my arm. Everybody asks me that. Yeah, I even have, I got all kinds of stuff. Anyway, my point is I'll do hashtag devil microbes and I'll look for people who find like microbes are terrible. Like all the time we get what I call swab stories where someone swabs an environment and guess what? There are bacteria there. Are you excited by that, Annelise? Are you frightened the way everyone else is? I call these swab stories. They make me angry, right?
And I have a favorite swab story.
Pardon me?
There was a Penn and Teller episode where they swabbed, like butts or something like that. And they were like, oh, there's bacteria on toilet seats, but there's more on your phone. And I'm like, but it doesn't really matter in either case until you know which ones they are. And anyway, swab stories. I love that. I'm going to keep that hashtag in my mind.
You're absolutely right. I would love to tell you it's original with me, but who knows? The other hashtag is angel microbes. And that's what you were talking about, where people say you can do, like, for example, there was some recent discovery about the gut brain and connection to the vagus nerve that takes place. And some people are claiming that certain lactic acid bacteria actually calm your mood. And when I say there have been studies, I always ask students to drill down into that, so to speak, and you'll find out it's not quite as clear cut as it's been presented. And because you knew what was going to happen. It happened within a year. People were trying to sell yogurt, claiming it would be able to treat depression, which is like offensive to me because that's a real problem, right? And I don't want someone who's dealing with that eating yogurt and feeling they've done something wrong where they don't feel better, right? But the fact of the matter is, and this speaks directly to what Annelise was talking about in her presentation, the vast majority of microbes are what I call meh microbes. They could care less about us. They really don't care. We're not even a surface to them. And we put all, and we're kind of, if you think about it, we're narcissists. So we're interested in the ones that do things to us, but they were here first. They really were. So that's what I would say there. And like with my centrisms, I talk about that with students, and I talk about the angel, devil, meh microbes. I even had an artist make me representations of all those things. But it's really true. One of the things that I do in my lab right now or my classes is we're swabbing reusable water bottles to show what lives there. Yeah, follow me on Twitter. You get to see all the pictures. It's a lot of fun. And people freak out like it has something to do with how clean they are. It has nothing to do with it. You're selecting, as Annelise would tell you, for biofilm formers, right? Yeah. And so we're doing some studies on that in my class as part of a cure. But I mean, you know, these things are happening, but people, and it's like the word germ drives me nuts. People automatically think it's negative, but they don't feel that way about wheat germ.
Or bug, right?
That's because Pasteur used the term germ to mean a piece of life when he was debating the spontaneous generationists and vitalists. That's all that meant. So here we go. And then I was doing so well getting people to not think that microbes were bad. And then this thing shows up.
Yep, for sure.
Yeah. Hard to overcome COVID.
It's the frowny face COVID.
So okay, both of you clearly have come to be able to preach the microbe, gospel here, and have overcome your eukaryocentrism. So I'm wondering if you could each talk a little bit about how that came about for you. Like, how did you come to think that microbes are so cool and want to spend a lot of time studying them? Annelise, do you want to go first?
Sure. Well, I don't know. I always was interested in like strange diseases, which doesn't exactly correlate with the microbes are good thing. But I think that sort of started things. And for a long time, I thought I didn't like biology because I thought biology was just memorizing like different parts of a cell on like a sheet that your teacher prints out for you. And I didn't realize like how much more there is to the field and how that's really not biology at all, I would argue, to a certain extent. And so I think it really started when I started working in lab, and we were talking about soil microbes. And I was like, whoa, I did not even know this was a thing. And so I don't know, I guess just sort of exploring things from there. I think for whatever reason, at least for me, it lends itself to sort of casual investigation of like, let me just go and read this one page on Wikipedia about this one thing or something like that. And so I think it just kind of evolved from the soil microbe stuff.
So to speak, right?
Yes. Yeah. Not
no, I love that. So as somebody who teaches, introductory biology, as like that is it's near and dear to my heart to hear you say that, that type of educational experiences is not helpful, right? Making people memorize things. We know that can turn so many people off, but yeah, getting to have those research experiences. Gosh, well, I'm so glad that you have been able to find research that speaks to you, and wow, amazing work that you're doing. Yeah. Mark, how about you?
All my life, I've liked weird things. That should not surprise you, right? I like strange things. I like the unusual. I always have. And when I was 10 or 11, one of my brother's good friends had muscular dystrophy. And he had a lab that his father had built for him in his garage. And we did all the amateur scientist stuff. Okay. And so I was his hands for several years. And I did a lot of stuff with him. And it made me know I wanted to be a scientist. I did a science fair project that was very important to me, but that didn't involve bacteria. Can you believe that? Until one time, look at you, Kelly. By the way, awesome glasses. I wanted to tell you that.
Thank you. Every week I get a new front thing that goes on.
I need to do the same thing. It's kind of Jeff Goldblum-y, but I'll go with it.
Nice.
So anyway, what I was going to say is that Terry, who was the friend of my brother's, was working on his master's degree when I was in middle school. And he brought me home a plate of Photobacterium phosphorium. And I took that into the dark and be able to see that beautiful blue light and understanding why it was blue, because that's the wavelength that transmits best through water, and how it worked long before then. It completely changed the way that I looked at everything. And people make jokes about being in the dark, but that's where I found out I like the strange things. And it works against you if you're not careful. You'll say, I'm not going to work on a simple to understand system, like Delivibrio, for example, rather than Salmonella or E. coli. But just as Barbara McClintock wrote, there was a book on Barbara McClintock called A Feeling for the Organism. I understand microbes. I understand why they do what they do. So I think maybe they've trained me, or maybe it's the microbes in me that are making that happen. But there's just something about the depth and breadth and diversity of the microbial world that's always fascinated me. And I would love to be speaker of the microbes, because that's kind of so important to me, because they're everywhere. Everywhere there's liquid water and any kind of energy gradient. And that's why I think we're going to find microbial life anywhere we find liquid water in this solar system. And that means underneath Europa. And so you're agreeing with me, Annelise. You're on the same wavelength. I think Enceladus has it. And I hope you will live long enough to see it, Annelise. I hope I do. But it's amazing what we're going to see in the future, because either they're relatives of ours, fascinating, or they're not related to us, which is even cooler. Kelly, do you know about the fungi that can live on radiation?
There are some growing in Chernobyl, right? Crazy.
And that's because of the single and double bonds next to one another, which you see in both heme and chlorophyll. And for the same reason, it's all about the electrons, Annelise, like you said, it really is. And they're able to convert some of that into energy. And Arturo Casadevall has done some great work on that. And I believe I wouldn't be surprised to see some of that stuff up the space station, wherever there's a little bit of water vapor and a little bit of gas.
I think the International Space Station definitely has problems with microbes. I believe I've read some stories about that.
Yes.
But so unfortunately, I think Scott and I both have to be somewhere at 5. So this is about the end of the fun that we get to have today. Both of these talks were awesome. And I would love some other time to get together and chat with you two about like space and microbes and more of your thoughts about that stuff, because I'm finding that endlessly fascinating, and the implications for whether or not we should be putting humans on Mars and what that's going to do to microbial communities. And anyway, so many.
Put me in on that conversation, please. Yes.
Yeah. But for now, I will remind our listeners and the folks who watch this. Well, I was going to say, and the folks that watch us on YouTube, that we have a YouTube channel, but probably most of the folks will be watching it on YouTube. Will have gotten there already. So we'll put this up on YouTube, and we send updates about future events through Twitter and through a Google Forms file that we link to on our Twitter account from time to time. And thank you so much, Annelise and Mark, for coming. This was awesome. I learned so much. It was so much fun. You've got me thinking actually about all the ecological stuff that I learned, how much of it I should be trying to apply to like predator-prey interactions and microbes, and how prey think about predator risk, how that relates to how complicated bacterial environments are and how much energy they should put into sensing. And anyway, I'm sure that I'll be thinking about this all week, if not longer than that. So thank you very much. This was a lot of fun.
It was a pleasure. Annelise, you were awesome.
Thank you. Yeah, thank you for having me. This is really fun.
All right. Thanks again.
Thanks. All right.
Bye,
everyone.
Bye.